Hi everyone, welcome again to the course of structural biology. We are continuing with our last module, structure based drug discovery. In the last class, which is the introductory class of structure based drug discovery, I talked about history, I talked about how drug designing developed, I talked about methodologies, but what I was trying to say is drug discovery have a history of long time, but with gradual invention of better techniques, better science from a random luck based or huge screen based discoveries now shifting towards logic based drug development. Today my discussion would be on logic based or rational drug discovery. So, I will talk about the methods for rational drug discoveries and especially the enhancement in the speed of those innovation with the incorporation or advancement of computation. So, as I told earlier, rational drug discovery have divided into major two part, ligand based drug discovery, where two major methodologies are QSAR modeling and pharmacophore development, whereas another is structure based drug discovery, where we have de novo drug designing, where the drug designing is happening from scratch without any prior information, and docking based virtual screening. Now, though we do all those divisions, we talk about those techniques, as like I told in many biology processes, like I was talking about when I explained the ab initio based packages, ITAZR. I have shown that ITAZR, though it is ab initio based programming, but it include statistical method, it include threading, it include energy minimization. So, it is a combination. Similarly, in the drug discovery, in the rational drug discovery, it is separated with the presence or absence of the structure of the macromolecule, but the methods mix up when we go for a innovation. So, keeping that in mind, I will talk about ligand based drug discovery, where there are two of them. One is quantitative structure activity relationships or QSAR. Quantitative structure activity relationship models are regression or classification model used in the chemical, biological and engineering. Like other regression models, QSR regression models relate a set of predictor variables x to the potency of the response variable y, while classification QSR models relate the predictor variables to a categorical value of the response variable. So, when I am talking about my definition of QSR is based on regression, and if you do not know about regression, regression is used in statistical modeling. Regression analysis is a set of statistical processes for estimating the relationship between a dependable variable, which also called outcome variable, and one or more independent variable, often called predictors, covariates or features. So, if you see the correlation here, the randomly oriented points, we have a linear line which shows that they have a tendency. So, basically if you look at conceptually how you look at a QSAR, QSAR is a initiative where you want to correlate your small molecule with the function. So, what I mean? So, suppose you have ethane and you change it to propane. So, one carbon change. How you develop a equation having all those factors with the, so there would be y equal to fx in this relation. So, I will talk more about that. So, QSARs are the mathematical relationships linking chemical structure with biological activity using physicochemical or any other derived property as an interface. Mathematical methods used in QSAR include various regression and pattern recognition techniques. Physicochemical or any other property used for generating QSARs is termed as descriptors and treated as independent variable. Biological property is treated as dependent variable. So, your descriptors would be coming from the property of the small molecule which would be related to the biological factors, biological properties. The descriptors are independent, whereas biological properties are dependent. As I talked about in terms of concept, the basic assumption is that there is a mathematical function of the chemical properties which is related to the effect. Effect means the biological activity. Thus, the effect called y is a function called f of the chemical property called x. Mathematically y equal to fx, but how to find this mathematical algorithm? How we could make the equation y equal to fx for a particular compound and its biological property. Typically, we use a number of chemical compounds with known values of the toxic effect. So, here y you could consider toxic effects. For each chemical compound, we calculate a series of parameters called chemical descriptors. Then we find an algorithm that provides a quite accurate value similar to the real experimental value, so that this could replace the experimental performance. The final step is to check if the so obtained algorithm is capable to predict the property values of other chemicals not used to build up the model. So, you have y equal to fx, right. Now, what you do, you take y as a property like toxicity of a small molecule, and then you have a equation which define, as I told, like if you have CH3 and it changed to CH3 CH2 CH3, how toxicity would be changed. So, you have known value, and you see the change and try to fit an equation. Now, when this relation is fixed by using many such, now you get a unknown one, apply that equation and see if it is fit. If it fit, you have a good QSAR. This last phase, what is the last phase? The final step is to check if the so obtained algorithm is capable of predict the property values of other chemicals. This is called the validation of the QSAR model. So, here based on the known data you develop a model and then validate it with a unknown chemical. The last phase, the validation phase, is very important. Indeed, it is very important to generate a model which is working not only for the chemical substances used within the training set, but also for the other chemicals, the unknown set, the not included training set. The challenge is to define the correct statistical properties of the model. So, basically you could take QSAR in a way you have the chemical compound, you could have make or you could have described many properties of the molecule, let us say solubility, let us say its reactivity, let us say its chirality. All of these are properties which could correlate to the biological activity. But instead of doing that, if you get numbers, that would be much more understandable way to relate them. That is what QSAR is doing. So, this is the QSAR model. You have the molecular structure, you have the relation of y equal to fx, you have to set fx, you know the activity, right. There is a structure and you know its binding activity. So, you have molecule A1, A2, A3, A4, A5, you know their structures and you know their binding affinity towards enzyme A. So, how? So, you have the molecular descriptors there describing the chemical property of this group of compounds and then there would be the binding affinity. You develop a relation between them which would come through linear regression, and then when you set up the model you validate it by bringing B. So, these are known for B, you know the structure, you determine using this equation the binding energy theoretically and then you do it experimentally its binding affinity and see these two are coming closer or not. If these two come closer, then your model is good. Molecular descriptors, as I told, simple rules for describing some aspect of a molecule, like you could talk about the structure, you could talk about the property. 2D descriptors only use the atoms and connection information of the molecule, like CH3, CH3. So, the atom carbon and 3 hydrogens, carbon and 3 hydrogens. If only the atoms and their connector, that is 2D descriptor. Internal 3D descriptors use 3D coordinate information about each molecule. So, it will show the 3D distance, angle, dihedral and all those information. Internal 3D descriptors use 3D coordinate information about each molecule. However, they are invariant to rotation and translation of the conformation. External 3D descriptors also use 3D coordinate information, but also require an absolute frame of reference molecule docks into the same receptor. So, internal 3D descriptors have only the 2D descriptor plus the 3D information, coordinate information. External 3D descriptor also needs the reference frame. Descriptor types. So, descriptors you already understand have a very important role in QSAR model, because you just understand the chemical molecule by the definitions or the descriptions coming from the descriptors. So, there are many descriptor families, constitutional or information descriptors which talked about molecular weight, number of chemical elements, number of hydrogen bonds or double bonds and many more. Physicochemical descriptors which talks about lipophilicity, polarizability and many others, like topological descriptors talk about atomic branching and ramification. Electronic, geometrical and quantum chemical descriptors are there. fragmental structural key. When you go for fragment based drug designing, there needed fragmentation, means you fragmentized a molecule, how the properties are changed. So, it is very critical how you select the descriptors. What is particularly relevant to the therapeutic target? If you ask somebody, is this person honest, and the guy reply, he is very good in academics. So, the question did not get answer, right. So, you have to choose proper questions. Sometimes a question is definitely a question, but the question is irrelevant in terms of what you want to know. So, how the descriptors would relate to the biological activity, your question selection should be dependent on that. What variation is relevant to the compound series, like you have, as I was talking about, ethane, propane, butane. What is varying here? A carbon, the topology, how those variations are related, you have to get those answers. What property data can be readily measured? What can be readily calculated? So, you have to understand those questions when you are going to select the descriptors. There are few assumptions when you perform QSAR model development. The effect is produced by model compound and not its metabolites. So, if we are thinking about a biological molecule which is involved in a metabolic pathway, we should know the compound we are targeting and not the transformed version. The proposed conformation is the bioactive one. The structure which you are selecting should have the optimized conformation related to the bioactivity which we are looking for. The binding site is same for all modeled compounds. If you are considering a series of molecule towards its binding to an enzyme or a protein, it should bind to exactly same biological macromolecule. The bioactivity explains the direct interaction of molecule and target. Pharmacokinetics aspects, solvent effects, diffusion, transport are not under consideration. So, how you make the model simplified depends on those assumptions. So, how you go for a model development? First, selection of training set. You will take molecules A1, A2, A3, A4, A5. You have to select them first. Enter biological activity data. Generate conformations. Calculate the descriptors. Then selection of statistical method. Generate a QSAR equation. Validate the QSAR equation. Predict it for unknown data set. So, that is the process for generating or working with a QSAR model development. Descriptor properties, it could have physical property like molecular weight, log P is permeability, permeability of a compound in octanol and water, because octanol is a organic solvent and water is a aqueous solvent. So, that is what we should calculate. Boiling point, melting point, dipole moment, solubility. So, all the physical properties. Structural descriptors. Structural descriptors are divided into two parts, 2D and 3D. In 2D, as we have explained earlier, you have to look at atom bond counts, number non-hydrogen atoms, number of rotable bonds, number of each functional groups, 2 carbon chains, 3 carbon chains, 4 carbon chains, 5 carbon chains, etc. Rings and their size. In 3D, number of accessible conformations which are biologically active, inactive, surface area. Topological description. Topological descriptors are defined by Wiener path index. What is Wiener path index? In chemical graph theory, the Wiener index, also called the Wiener number, introduced by Harry Wiener, is a topological index of a molecule. It is defined as the sum of the lengths of the shortest paths between all pair of vertices in the chemical graph, representing the non-hydrogen atoms in the molecule. Like if you have all of them carbon, how they are connected, one connected directly to 2, 6 is not understood, but just one atom away from 1. So, this is the relation and this could be calculated, and when boiling point for alkanes are considered. So, this is the in the y axis is the boiling point and the x axis is Wiener index. You see that there is a very nice correlation. So, you could successfully use topological descriptors using Wiener number with the biological activity of the molecule. Type of QSARs, 2-dimensional QSARs like classical Hunt's analysis, 2-dimensional molecular properties, 3-dimensional QSARs, 3-dimensional molecular properties, molecular field analysis, molecular shape analysis, distance geometry, receptor surface analysis. So, let us look at a case study, receptor surface model. Hypothetical receptor surface model constructed from training set molecules 3D shape and activity data. The best model can be derived by optimizing various parameters like atomic partial charges and surface fit. Descriptors like van der Waals energy, electrostatic energy and total non-bonded energy can be used to derive series of QSAR equations using GPLS statistical method. And when you do that, you see this is a graphical representation of the receptor surface generated around the training set of molecules showing steric and electrostatic fields. So, by generating this, you could describe, you could develop a much better model from the original structure, which would be beneficial towards its further interaction with biological macromolecules. Advantages and disadvantages of QSAR. Advantages of predicting biological activity with quantitative structure activity relationship modeling include: it able to predict activities of a large number of compounds with little to no prior experimental data on activity. So, we will talk about later one of the biggest problem which we also discussed in the previous class is that modern day drug discovery takes a lot of time and more importantly takes a lot of money. So, using QSAR, it is possible to predict activities of a large number of compounds with little to no prior experimental data on activity is extremely beneficial towards drug development. It can reveal which molecular properties may be worth investigating further. It regarded as a green chemistry approach since chemical waste is not generated when performing in silico predictions. In vivo and in vitro experimentation can be very expensive and time consuming. And if you now understand the process, I just include one more advantage. See, here we have the training set. the training set habits descriptors. You relate that to the biological activity and you develop the equation. Then you validate it with unknown and then use for prediction, right. If you have understood what I talked about machine learning before, this is somehow ready to go for machine learning. So, in current era where machine learning is coming up, contributing in every field, this would make QSAR models even better, and with more data coming here, the program would be automatically updated, and this is really wonderful because we are not talking about biological macromolecule here, we are talking about small molecule. We will get a lot of small molecule around, and using them the program can easily be upgraded. Also, QA modeling reduces the need for testing on animals and or cell cultures and save time. So, you get already a correlation, so you get selectivity. Disadvantages of QSAR: does not provide an in-depth insight on the mechanism of biological action. Some risk of highly inaccurate prediction of pharmacological or biological activity is always there. Sometime apparently there is no correlation. So, that is the risk fact. Coming to the second one for ligand based drug designing, the pharmacophore development. A pharmacophore, as you see a representation of a pharmacophore, is an abstract description of molecular features that are necessary for molecular recognition of a ligand by a biological macromolecule. IUPAC defines a pharmacophore to be an ensemble of steric and electronic features that is necessary to ensure the optimal supramolecular interactions with a specific biological target and to trigger its biological response. So, if you see, I talked about this, but I did not explain it. What you see here, the sizes and the charge. So, size and charge are the major factor of a small molecule to predict how it interact with a biological molecule, and that is what pharmacophore is doing. A pharmacophore model explains how structurally diverse ligands can bind to a common receptor site. Furthermore, pharmacophore models can be used to identify through de novo design or virtual screening novel ligands that will bind to the same receptor. So, basically what we are doing when you look at a molecule, like you are looking at this molecule, you look at it at 2D, you do not see the charge. So, it is difficult for you to fit it in the active site, the binding site of the biological target, but now you represent it in terms of its 3-dimensional size and charge. So, you could know its conformation, and it is much easier to predict its binding capability in a binding site. Pharmacophore features. Typical pharmacophore feature include hydrophobic centroids, which you see, aromatic rings which will be involving in base stacking and all, hydrogen bond acceptors or donors, cations and anions. These pharmacophoric points may be located on a ligand itself or may be projected points presumed to be located in the receptor. The features need to match different chemical groups with similar properties in order to identify novel ligands. Ligand receptor interactions are typically polar positive, polar negative and hydrophobic. A well defined pharmacophore model includes both hydrophobic volumes and hydrogen bond vectors. So, you get the size, 3-dimensional volume, you get the direction of the hydrogen bond donation or acceptance, you get the charges. Pharmacophore concept: signification of the spatial arrangement of a small number of atoms or functional group. You have a ligand, you have a small molecule which is apparently a little big, and in between there is a architecture. You are not bothered about that architecture, you are just bothered about the representative charges, how they are represented and what is the volume so that you could fit. So, you are actually pointing out the important parts of a small molecule which might play critical role in interaction. With the model in hand, search databases for molecules that fit this spatial environment. So, when instead of taking a complete molecule, you will take the pharmacophore model, you get to identify the similar type of interactor, similar type of 3-dimensional volume containing molecules. So, it would be great. It is a simple model I put here to enhance your concept. So, first you identify the binding site in the macromolecule target, then you identify the docking site where the possibility of hydrogen bonds which are critical for the interaction. You take out the hydrogen bond possibilities, like these are your pharmacophore, and based on them you develop possible leads and you use those leads to search for further libraries. Pharmacophore descriptors: number of acidic atoms means negatively charged atoms, number of basic atoms means positively charged atoms. So, if you consider amino acids, these are aspartate and glutamate. These are arginine, lysine, apart, histidine. Number of hydrogen bond donor atoms, number of hydrophobic atoms, sum of van der Waals surface area of hydrophobic atom means hydrophobic patches. So, here are some pharmacophores and you see that depending on how they interact, it is considered as 3-point or 4-point pharmacophores. How you develop a pharmacophore model? The process for developing a pharmacophore model generally involves the following steps. Select a training set of ligands, you get a set of ligands. Choose a structurally diverse set of molecules that will be used for developing the pharmacophore model. As a pharmacophore model should be able to discriminate between molecules with and without bioactivity, the set of molecules should include both active and inactive compounds. Okay. Conformational analysis: generate a set of low energy conformations that is likely to contain the bioactive conformation for each of the selected molecules. So, what you do? You select the library and go for energy minimization. Energy minimization will give you low energy conformations. Molecular superimposition, superimpose all combination of the low energy conformations of the molecule. Similar, which is called bioisosteric functional groups common to all molecules in the set might be fitted. For example, if they have a phenyl ring, all the phenyl rings come and like be there close to each other, or all the acid groups might be the compounds are different, but those points should be fitted together. The set of conformations, one conformation from each active molecule that results in the best fit is presumed to be the active conformation. Abstraction, transform the superimposed molecule into an abstract representation. For example, superimposed phenyl rings might be referred to more conceptually as an aromatic ring pharmacophore element. Likewise, hydroxyl groups could be designated as a hydrogen bond donor acceptor pharmacophore element in that way. Validation, a pharmacophore model is a hypothesis accounting for the observed biological activity of a set of molecules that bind to a common biological target. The model is only valid insofar as it is able to account for differences in biological activity of a range of molecules. So, basically there are so many ligands, they are diverse, but they are representing their functional group which could have the ability to bind to the receptor, and then if it correlate with the biological activity, then the model is developed. As the biological activities of new molecules become available, the pharmacophore model can be updated to further refine it. More and more biological activity correlating to small molecules should be available, updated better model could be obtained. Model example, I talked about how, but now you see the model. An example of a pharmacophore model of the benzodiazepine binding site on the GABA receptor. White sticks, if you see the white sticks represent the carbon atoms of the benzodiazepine diazepam, while green represent carbon atom of the non-benzodiazepine CGS 9896, another compound. Red and blue sticks are oxygen and nitrogen, as we know conventionally, that are present in both structures. The red spheres labeled H1 and H2A3 are respectively hydrogen bond donating and accepting site in the receptor, while L1, L2 and L3 donate lipophilic or hydrophobic binding. So, these are contributing to hydrophilicity, whereas these three contribute to hydrophobicity. How you search the 3D pharmacophore? With the pharmacophore in hand, search databases containing 3D structure of molecules for molecules that fit. You could rank these hits using scoring system, which would be, you have to describe it according to how you want it. Application of pharmacophore model. In modern computational chemistry, pharmacophores are used to define the essential features of one or more molecules with the same biological activity. A database of diverse chemical compounds can then be searched for more molecules which share the same features arranged in the same relative orientation. Pharmacophores are also used as the starting point for developing 3D QSAR model, because they define the properties Now, you use those properties, develop descriptors, correlate it, and now get a new QSAR model. Such tools and a related concept of privileged structures, which are defined as molecular frameworks, which are able of providing useful ligands for more than one type of receptor or enzyme target by judicious structural modification, would aid in the process of drug discovery. So, with that we are coming to structure based drug discovery. The performance of biochemical processes and cell mechanisms are dependent upon complex and multiple non-covalent intermolecular interaction between proteins and small molecule modulators. The understanding of the structural and chemical binding properties of important drug targets in biologically relevant pathways, which allows the design of small molecules which are capable of regulating or modulating specific target function in the body that are closely linked to human diseases and disorders, which is our target. This is mostly done through multiple intermolecular interaction within a well defined binding pocket. So, we look at a biological macromolecule which is a target, and we generally try to get the binding pocket, which in case of enzyme is the active site pocket. In general, the identification of promising hits for further optimization is a major challenge faced by the both pharmaceutical and academic laboratories. Although the trial and error nature is inherent in drug research, rational concepts are always more acceptable way. But going rational, utilizing structure related information would be successful only if modern computational methods have become widely employed for lead selection and optimization. So, you use structure, you get high resolution structure, but you have to use it to calculate by fitting with many, which needs the computation, the algorithms as well as the ability to do compute. In other words, what we normally call structure based drug discovery could be very well termed as computer assisted structure based drug discovery. The use of three-dimensional protein structure information in the development of new biologically active molecules is termed as structure based drug discovery, or SBDD, is a well established, successful and highly attractive strategy used by academic and pharmaceutical research laboratory worldwide. As a creative and knowledge driven approach, an essential requirement for structure based studies is a substantial understanding of the spatial and energetic aspect that affect the binding affinities of protein ligand complex. So, basically as we started and talked continuously, drug designing is about the macromolecule and its interaction with small molecule, peptide or macromolecule. But we are now mostly focusing with the small molecule and peptide. Considering that the shape and chemical nature of the binding site of a specific target protein are known, along with the possible intermolecular interaction between ligands and the protein within its active site have been identified. So, it is a very good point. You want 3D structure, but if you get a 3D structure of the protein in apo form, it is good, but it is not as good if you get it bound to the ligand. So, this will give you the set of interaction between the target molecule and the potential substrate or inhibitor would make your life more facile to work further from this existing foundation. We could use those information directly for the identification of new ligands and the optimization of lead compounds. This opens new possibilities to boost the search for lead molecule and to limit the number of compounds that need to be evaluated experimentally. Hits can be identified through the docking of small molecule ligands selected from database of chemical structures into protein active sites or by using receptor based pharmacophore models. Furthermore, drug candidates can be designed de novo by improving the complementary binding properties of the lead molecules. It could also improve by understanding the respective target proteins, that is the intermolecular interaction between the amino acid residues of the target active site and the chemical groups. So, if you have these, you know that there is a oxygen. So, how the interaction will happen? Is it hydrogen bond? Is it charge interaction and all those things? Molecules that mimic the transition state of enzyme catalyzed reaction are interesting example. I will talk about these in the case study, but just to explain little bit. Transition state. What is a transition state? I talked about this, but like AB when react with CD will form AD plus BC. So, this is called substrate. These are called product, but in between there is a situation where the bond between A and B are going to be broken and the bond between C and D are also going to be broken. At the same time, there will be bond between A and D going to form, there will be bond between B and C going to form. This situation which exists theoretically, but does not exist practically is called transition state. Now, why transition state of enzyme is important? Because in transition state the enzyme generally shows its intrinsic property, and you remember it is very difficult or nearly impossible to change your intrinsic property, and that is why if you somehow make the stable molecule which mimic this transition state, they are always good drug. I will talk about them. Early drug discovery step usually requires structural optimization of lead compounds in order to build the highest possible level of potency, selectivity and affinity for the target of interest. It would also require appropriate physicochemical and pharmacokinetic characteristics. Substrates and cofactors of several enzymes have been structurally modified to generate excellent inhibitors using X-ray crystallographic data, because that gives you the highest resolution, though cryo-electron microscopy is surprisingly going higher to higher level of resolution, and now they are providing atomic level resolution in a regular basis, which I have discussed earlier. So, the methods of rational drug discovery, as I talked about the structure based drug discovery, there is docking based virtual screening and de novo drug designing. Today we will talk about de novo drug designing. De novo drug designing means start afresh from the beginning, from the scratch. So, if you start designing a drug from scratch, that unique process is called as de novo. Now, you will ask me, if it is from scratch, why it come under structure based drug discovery? Yes, I am going to give you the answer. So, it is a process in which the 3D structure of biological macromolecule, usually protein, is used to design newer molecules. So, de novo drug designing is a scratch from the side of the small molecule, but there is must existence of the 3D structure of the biological target. It also involves structural determination of the lead target complexes and lead modification using molecular modeling tools. Information available about target receptor, but no existing leads that can interact. Build compounds that are complementary to a target binding site on a protein via random combination of small molecular fragments to make complete molecule with better binding profile. So, you see, this is the initial core fragment and this is the biological target. The fragment to be added is this. So, initially you see that there is a interaction possible. This is the first step. Then you include this and you optimize. So, in the step 2, you add a fragment and optimize its conformation. This is wrong, this is right. And now you have the lead compound. So, you could have receptor based design or pharmacophore based design. So, you have pharmacophore based design which is applied to receptor based design, you have receptor based design which will go for pharmacophore based designing. Can pursue both receptor and pharmacophore based approaches independently. If the binding mode of the ligand and target is known, information from each approach can be used to help the other. Ideally, identify a structural model that explains the biological activities of the known small molecules on the basis of their interaction with the 3D structure of the target protein. Steps of de novo drug designing: generation of potential primary constants, which is the first step, derivation of interaction sites, building up methods, assay or scoring, search strategies, secondary target constants. Okay. These are the 6 steps. So, first you do the generation of potentially primary constant, which I saw. You bring a fragment and see the interaction with the structure of the biological macromolecule, then you derive the interaction site, you build up by linking others, then you do the assay and you get the molecule, use as a lead, you do search strategies and then check for other, means check for side effects. Primary target constants: these are the molecules which set up a framework for the desired structure with the required ligand receptor interaction. So, you have it and you have a group here. Based on this you do complementary identification. These are of 2 types, one receptor based, which is interaction of the receptor from basis for the drug designing, ligand based, ligand to the target function as a key. Derivation of interaction site, a key step to model the binding site as accurately as possible. This starts with an atomic resolution structure of the active site. As I told, structure is must. Programs like UCSF doc define the volume available to a ligand by filling the active site with spheres. Further constants follow using position of hydrogen bond acceptor and donors. Other docking algorithms such as FLOG, GOLD and FlexiDoc 16 use an all atom representation to achieve fine detail. Ray tracing algorithms such as SMART represent another strategy to filling up. Building up methods. Building up methods, there are 4 types: growing, linking, lattice based sampling and molecular dynamics based methods. I am not going into all the details, but growing means, as I showed, it would grow gradually by coming up with different ligands and how one fit. Linking, it comes with fragments. You have a library, there are, let us say, 2-3 binding sites are there, or part of the binding sites are there. So, 3 fragments bind there and then do the linking. Lattice based sampling is another one. The whole lattice would be created, and after growing and linking it will fit to the lattice and lattice based sampling would be performed. Molecular dynamics based method, this is computationally intensive. We will talk about this more on the next class, but just to, it would use molecular dynamics based energetics. There are 3 other methods: assay or scoring method, search method and secondary target constant. All of them I will discuss in the next part when we are explaining the docking based virtual screening. There are other programs available for de novo drug designing. For site point connection method, there is LUDI. The fragment connection method, there is splice, new lead, proligand. Sequential build up method, legend, grow, sprout. Random connection and disconnection method, concepts, concerts, McDor LG, MCDNLG. Coming to the summary, in de novo design, the structure of the target should or must be known to the high resolution and the binding to site must be well defined. This should defines not only a safe constraint, but hypothetical interaction sites typically consisting of hydrogen bond, electrostatic and other non-covalent interactions. This can greatly reducing the sample space, as hydrogen bond and other anisotropic interaction can define specific orientations. Although a relatively new design method, de novo design will play an ever increasing role in modern drug designing. Though yet not able to automatically generate viable drug by itself, it is able to give rise to novel and often unexpected drugs. So, it is always good to go for this method if you want some magic. Relatively slow and inefficient, but when coupled with high throughput screening is proving to reduce drug designing turnaround time. Ignores synthetic feasibility while constructing structures, which is a very good thing. Considering current scenario, this technique cannot be a sole basis for drug designing, it is dependent on the other techniques. The number and variety of structures which could be identified are limitless, and so the chances of hitting the ideal structure is normally poor. There is far more to drug designing than finding structure that binds strongly to its target. It does not identify whether the structure identified will have favorable pharmacokinetic properties or acceptable safety profile. So, the process of de novo is not at all connected to identifying a compound which did not have other biological side effects. It can simulate new ideas and identify novel lead structures which could then be optimized through rational drug designing. So, today I have talked about a description of QSAR, how the models developed, how QSAR work and what are the application. Then I talked about pharmacophore modeling, and then I talk about the details of some structure based drug discovery and how a very newly upcoming method, de novo drug designing, is working. Thank you very much. You guys are amazing listener. Keep asking questions if you need. Thank you.